I can add these bacteria to rice and corn and tomatoes and bell peppers and even Douglas fir to increase plant growth and health without having to use chemical fertilizers, pesticides, even water. My dream is that we can drastically reduce the amount of nitrogen fertilizer we use. It's causing so many environmental problems.
When you see an area like this, you see mostly trees, but it's a complex, interrelated community of many organisms.
Some of the most important processes are driven by microbes and therefore hidden from our views.
Imagine if we can use these microbes to grow plants in arid regions, marginal soils, and even contaminated soils.
And that's why we're here, just outside of Seattle, to meet Dr. Sharon Doty, who's using genomic technologies to make this dream a reality.
Why are you collecting samples at this location?
If you look below you, you're standing in rocks and sand, and yet these trees are flourishing here. What led me here originally was I was doing poplar tissue culture, and there was always this contaminant in the way that was this slimy bacteria. I found out it was a rhizobium.
You usually think of these things as growing in nodules.
Right, yeah, only in root nodules of legumes or Frankia in alder trees. And so then I thought, well, if there's rhizobium in the poplar tree, maybe it's getting nitrogen from the bacteria. I made cuttings, just stuck them in nitrogen-free medium, and sure enough, the trees were totally healthy.
It's rhizobium in these trees as well?
There's some rhizobium, but it's mostly other species. There are lots of Burkholderia, Herbisporillum, Pseudomonas, and a lot of the bacteria can fix nitrogen.
During nitrogen fixation, the oxygen inhibits the process. How do the plant manage it? Because we always thought that the root nodules were critical to that whole process.
Yeah, that's a dogma I've been battling against for many, many years. It's true that nitrogenase enzyme is inhibited by oxygen, but they're free-living bacteria that can fix nitrogen. So they're not waiting around for a plant to make a nodule for them to use this enzyme. It's really not a problem. It's only a problem with the dogma. So taking the branch samples that we collected at the Snoqualmie River site, I cut it into little pieces, then I surface sterilize it, and then grind it up using the pestle. We can't do it using liquid nitrogen because that would harm the microbes. So I just grind it up until I have this nice green juice, so we call it a poplar smoothie. We purify the strains and we characterize them, and then have the full genome sequenced, and then we can expose the poplar roots to this solution, usually for a day, and then we put the plant on selective medium. Usually we're making a whole consortia of different strains, having a lot of potentially symbiotic properties, including nitrogen fixation. We can fluorescently tag these microbes and follow them.
It is so weird to see the microbes inside the plants. How do the microbes actually manage to get into the plant?
We've shown that they can enter into the plant through these lateral root junctions. So it's called crack entry. The defense responses go up initially, but then that response goes down.
And do the microbes stay?
3 years later, I was able to re-isolate that same bacteria from those trees.
And this colonization is even, or is it kind of clustered?
We found that the microbes form these microcolonies within the plant, and we think that that might be where they're fixing nitrogen.
Can you take this technology and move it to other plants?
We've shown again and again in so many different plant species that we can add these microbes, and then they have these ability now to grow with limited fertilizer. We've also found that the microbes have all sorts of other benefits to the plant. So it's overall plant health and stress tolerance.
Are there certain strains that actually work better, or is it something that they need to breed back in again?
Well, I think you're right that they've been breeding all of these agricultural crops for their response to chemical fertilizer. And now we need to start breeding for the ability to interact well with these beneficial microbes.
There are a few key processes. For example, photosynthesis, transpiration, or respiration. All these impact the plant physiological functions and therefore the crop production. First, I inoculate the plants. About 4 to 5 days after they're germinated, I measure the photosynthesis using some hand-to-hand instruments. We hope to see this symbiotic technology impact producing agricultural crops in a better way, using less resources to grow the plants in the agricultural ecosystem.
A strong area of research in my lab is called phytoremediation. That's using trees to clean up environmental pollutants.
In Tacoma, there was a smelter that operated for about 100 years and has polluted large areas, about 1,000 square miles of Puget Sound, with arsenic, cadmium, and lead. The challenge with heavy metals or metalloids like arsenic is that they don't break down or they don't degrade in the environment. The Asarco site itself fell under an EPA Superfund site, and so it was remediated using conventional remediation technology. Outside of the Asarco site are thousands of houses that have arsenic contamination, and so we think that using phytoremediation, something that could extend the reach of the conventional remediation activities. We want to have plants that move the arsenic from the soil into the roots and then into their above ground tissues. We're looking at woody plant species like willow and poplar, which can be coppiced, which means just cutting them off at the base and then removing the plant tissues from the site. And then they'll regenerate or regrow off of their roots. And so it's an annual product that you can just harvest the above ground tissues and remove the arsenic off-site.
What was the biggest surprise you've got from the work you've done?
Just finding out how many different ways that these microbes are impacting the plant growth and plant health.
How is that going to impact the future?
Yeah, I think it has huge implications for every time that we grow plants, whether it be for agriculture or forestry or bioenergy, that now we have the means to make it more not only environmentally sustainable, but also economically sustainable.